everyone. Welcome to the final lecture in the series for thorax. This one will be going into the heart, which you could sort of say is the heart of the series. Again, looking at the objectives, let me know if you have any questions. Hopefully they're pretty straightforward, but let's get right into it. Okay, so let's get into the pericardium. We mentioned the pericardium when we were talking about the mediastinum, particularly the middle mediastinum. It was one of the organs we mentioned. And the pericardium is basically like a cavity that contains the heart. Now there's two separate parts. We have a fibrous pericardium, which is going to be this nice little structure on the outside here. And then we have the serous pericardium, which is basically going to be the same thing as our pleura in the lungs, which means we're going to have a parietal pericardium and a visceral pericardium. And those are going to work exactly the same way, remember? So visceral means it's going to be on the organ, so the visceral layer will be on the heart, and the parietal layer will be lining the inside of that pericardial sac. So if you were looking at a body for this, and you look at some of the dissection videos, you can see how thick this sac is. And it really just, the goal is to, again, give that little serous fluid layer, so as the heart is pumping blood, the heart is beating and pumping blood rapidly, and hopefully not too rapidly, it doesn't build up a lot of friction. It can just slide along that pericardial layer. So here's kind of a nice close-up diagram. The parietal layer is going to be nice, adhesed to the fibrous layer, and the visceral layer we actually refer to as the epicardium. It's going to be a single cell layer, and here, if we were looking at the microscopy of it all, we have a myocardium, an epicardium, and then the inner lining is going to be an endocardium. So the outer lining, if we were to look at a cross-section of the heart under a microscope, is actually part of the visceral layer of the pericardial sac. And is a composition of the wall of the heart. So here you can see like a nice in situ image. So you got the nice fibrous pericardium on the outside. Everything on the actual heart is going to be serous pericardial, and then the lining on the inside of that fibrous pericardium is the parietal layer of the pericardial. All right, so let's take a look now at the fibrous pericardial attachment to the diaphragm. So we had the central tendon of the diaphragm, which is going to be a nice adhesion point for a lot of structures. And what you'll see there, if you were actually trying to remove the heart you can pull out of the pericardial sac, but the pericardial sac actually adheres really tightly to the diaphragm down here. And we call this the pericardial cophrenic ligament. So remember, we saw the pericardial cophrenic arteries, which are basically going to be tied to this pericardium, diaphragm region. So pretty much think of anything like that. If we break down the words, peri, around, cardio, heart, phrenic is diaphragm. So it's everything kind of around that area that's being supplied and attached to the diaphragm here. If we're looking at blood supply, as you might imagine, the pericardiophrenic arteries, pericardiocophrenic arteries are going to be supplying most of the blood to the pericardium. And then also that musculophrenic, which is going to be part of the terminal branches for your internal thoracic artery, is going to continue to supply after the split at about the sixth rib. You'll also get supplies from the coronaries. So as the coronaries come off and supply the the visceral pericardial sac, some of them can also apply a little bit of the parietal layer as you get closer to the posterior aspect of the heart. Nerve supply, pericardial nerve supply comes from the phrenic nerve. So we saw that as some of those sensory fibers are coming off. The only motor innervation from the phrenic nerve is the diaphragm. All other sensory is going to be from the pericardium, as well as some of the diaphragm and some of the parietal pleura as well. So here you go, you can see our phrenic nerves. If you remember, those are going to be part of our middle mediastinum. They're just kind of hanging on the side there, so you should just sort of see those dangling down and getting close to the heart in that region on the left and right side. So they'll actually be in kind of suspended there. So if you were to do a dissection in this cavity, they can be easy to split because they're just sort of, especially when you're trying to separate them from things like the pericardiocophrenic or the internal thoracic artery. But just be mindful that you should be able to see any sort of, if you see the nerve next to the heart, think phrenic nerve. The vagus one is often confused, but that one's going to be a little more posterior, and that's going to be, especially on the left side, easier to see because it's riding on the back of that aorta, or the front, I should say. All right, so when we break down what the actual heart is, that was all pretty much pericardium. That's just the stuff that houses the heart. The heart is really just a pump. It's a glorified pump, a huge sac of muscle. It's the only portion in the body that has cardiac muscle, very specialized muscle tissue for this. Involuntary muscle that is also striated. But it's got 4 chambers, and the goal is just to get blood to the rest of the body. And so we want to make sure that it's all working in sync and as it should. But yeah, it's just a hollow pump that basically just kind of keeps moving things through. So let's talk a little bit about the blood flow. So after everything has come from the body and it has entered the cable system, and when I say cable, it's either inferior vena cava or superior vena cava, it's all going to dump into the right atrium. That's going to be all deoxygenated blood, and that's just going to basically arrive here. You can see that deoxygenated blood, they do the classic coloring in purple. And what's going to happen is then it's going to get pumped into the right ventricle, and the right ventricle is going to pump that into the pulmonary arteries. So those are the only arteries with deoxygenated blood, where it can get oxygenated by the lungs. So the lungs are going to bring it back by the pulmonary veins and dump it in the left atrium. So atriums are always just going to be the housing dump areas for all the blood that's returning to the heart. The ventricles are going to be the muscular components that do all the pumping. So you go and you wait in line at the atrium, you get approval to go into the ventricle, and then it will send you off to where you need to be. So from the left atrium, this well-oxygenated blood is going to drain into the left ventricle. Left ventricle tends to be more muscular, and it's going to pump it through the ascending aorta, through the arch, and start filling up all the other areas of the body that need the blood. And so that's why we want to make sure our left ventricle is nice and thick. Not too thick, but thick enough where it can use that muscle compression and create a good pressure for the rest of the body. So when we talk about the cardiac cycle, we refer to it as having diastole and systole. So diastole is when you have the ventricles basically just filling up. It's kind of like a resting heart part of the beat. So the pulmonary valve and the aortic valves are going to close. The AV valves, and we'll take a look into those in a little more detail shortly, are going to open and basically allow blood to go from the atrium into the ventricles. So diastole you can sort of think of as like a resting heart. It's basically the heart is just pumped, and then it kind of expands and gets ready to pump more blood. So that's diastole. Systole is when the heart's actually pumping. So here you can see our blood that's gone into our ventricles basically gets pumped out. It pushes it through our aortic and pulmonic valves, and this mere act of pumping is going to be what closes our AV valves, our atrioventricular valves. And we'll see those they have a special mechanism. I find them kind of fascinating. Actually, both types of heart valves are kind of fascinating in their own way. So that's your basic heart function. When we look at the heart, I kind of mentioned this before, you break up the heart wall into 3 layers. We have an endocardium, which is basically like the inner layer. So if you think about it, the heart is actually kind of like a big blood vessel, but a little more complicated than that. And the blood vessels had 3 layers, just like the heart. So we had the inner cell lining, which is like the endothelium. In the blood vessels, we have the endocardium. You have the myocardium, which is kind of like the media layer of your blood vessels. And then you had the epicardium, which was the adventitia. And in our case, it's going to be the visceral layer of the pericardium. So all your musculature is going to be in the myocardium. The other 2 layers are thin layers of just single layers of cells. So the other thing to note is the heart, in order to maximize its contraction, and I'll have to see if I can find a really cool video of someone, you can actually dissect a heart and just kind of roll it out like a paper towel based off the sort of organization of the muscle fibers here. So if you think about it, let's say you go, you take a shower, you go swimming or something like that, and your towel is soaked. You know, what is the way we usually drain our towel and wring it out of water? Is like we twist it, right? So you just kind of like twist it, and that sort of creates an extra bit of pressure to get out any excess, in that case, fluid. And the heart works very similar in that regard. So what you want to do is have these concentric rings so that when the heart pumps, it doesn't just squeeze, it twists, okay? And that twisting is what's going to give it an extra oomph and get that blood out of there and make sure your pressure is sufficient. The heart does have a skeleton. So we refer to this as a fibrous skeleton. It's got basically the necks of the, I should show it on here. Basically, it has the outline for where your cardiac valves will be. And then you also have these little trigones in the center that are going to hold those pieces together. But this annulus ring, this fibrous ring, is basically your heart skeleton. It creates a anchoring point for our valves and makes sure those are kind of in sync, and then all the musculature attaches around it. So nothing too special about it other than the fact that it's just there, and it kind of helps. You'll see that if you were to look at a heart structure, kind of the nice shapes of the valves. When you open those up, they kind of have a nice holding of their structure, which is kind of cool. As far as where the heart lies, so typically in your classic presentation, the heart is going to dip to the left side of the body. So we say it rides 2/3 left of the median plane. So the median plane is the one that's directly in the middle. And then you only have 1/3 on the right. So that's a classic example where a heart is on the left side. Now you can get variations just like in anything else. One of the more common ones is called Dextrocardia, and it's where your heart actually sits on the right side. Basically same thing, but it's on the right side. That one's usually associated with a lot of issues, particularly things like transposition of your great vessels, which basically just means your pulmonic artery and your aorta are switched and doing each other's jobs. But less commonly, which is a little more stable, is what's called Situs Inversus. So this is kind of a cool thing where your whole internal organs are flipped. So your lung is actually, or sorry, your liver's on your left side, your heart is dipped to the right side, and everything works fine. It's just kind of flipped. It's a mirror image of what a normal body would look like, which is kind of interesting. If you don't know you have that, then you go to a doctor and they're trying to listen for heart sounds. They can't because, you know, they're listening to the wrong side. All right, so let's take a look at the surface anatomy of the heart. So we have an anterior surface, which we refer to as a sternocostal surface, basically meaning it's on the, it's abutting the ribs and the sternum. And this is mainly going to be your right ventricle. So everything you see here is predominantly right ventricle. You can see our pulmonary artery here. So most of the anterior heart is right ventricle. You have your apex. And then you have your, we'll just go into the left pulmonary side, which is going to be mostly left ventricle. So everything over here, so you can see there's going to be, if you were to palpate, there's a septum here where our vessels ride, and then over here is going to be our left ventricle. We have the right side, which is going to be our right pulmonary surface. This is pretty much all right atrium. And then you have the inferior surface or diaphragmatic surface. So the heart is actually kind of switched or flipped a little bit. So our left ventricle is going to be abutting our left lung, and it's also going to be kind of on the inferior aspect of the heart, where our right ventricle is a little more anterior superior, so it's going to be above that. So that's just, the heart doesn't just kind of sit there and hang into our chest cavity. It actually is a little more slanted, and everything like the right side stuff, the right deoxygenated area is more superficial, and then the left side is a little more deep, and it tends to be a little more broader. So just be mindful of that. The apex is the one that, so again, in a typical heart, we usually see that extending to about the 5th intercostal space. So you can actually kind of palpate that if you were to palpate where your ribs are. If you go to the 5th intercostal space and they want to auscultate where your heart sounds are, they actually can use that and find the apex and see, they can test the various locations for your heart sounds. So the apex is basically the tip there. And the thing to make note of in this image here, let's see if I can point it out, expand a little bit. So right about here, let's see, right about here, we'll get there. This is our intraventricular septum. And so this is the dividing point. So our left ventricle is kind of just starting at the tip, and you can see where the septum, and the easiest way to find this is look for the vessels. And so this is all left and this is all right. So our apex is predominantly part of the left ventricle. That's not 100% true all the time, but you can see where if we were to just straight cut this and think we would see both ventricles, we actually probably wouldn't in this case because the septum is actually kind of deviating a little bit more inferiorly. What I want to do is I actually can probably put together a little video for heart anatomy as well. All right, so when we refer to the base of the heart, that's the back side of the heart. So predominantly that's going to be your left atrium. It's where a lot of all our pulmonary veins are going to come in. And we can see those are paired on either side, right? So they're all dumping into that left atrium. We'll also see our pulmonary trunk split into our left and right pulmonary arteries. And then you'll also see the coronary groove for our veins are going to be located on the posterior aspect. But if we're looking posteriorly, this is all left atrium. And then we already talked about how this is pretty much all left ventricle, and then the left ventricle is going to be on the underside too. All right, so again, going back to the dynamics of blood flow, right ventricle is going to receive, or right atrium is going to receive the blood. And what we see in the right atrium is it's going to have an auricle, which is basically like, it translates to literally ear. It's kind of a vestigial organ here that you can see on both the left and right side. And it's going to basically hug the aortic, ascending aorta. And that's a nice little landmark because it helps you find where your right coronary is, because usually you have to lift that to find the right coronary artery under there. So if we're looking on the inside, so we have a region called sinus venerum, which if you were to open up, there's this smooth portion of heart muscle, which is sinus venerum. The roughened portion, which you can kind of see over here, is referred to as pectinate muscle. Now, that's something that I always like to test people on, because we'll see the roughened patch in the ventricles is referred to as the Trabeculae carinae. So only in the atrium do you have pectinate muscles, and mostly in the right ventricle, but we will see those in the left, because all the auricles of the heart will be full of pectinate muscles. So that's kind of your basic premise. The sinus venerum is sort of a developmental structure remnant from where the vena cavas go, and they help sort of funnel the blood into the, getting the blood into the right ventricle. And part of that is an issue from when we had a communication between the left and right side through the foramen ovale, and we'll talk about that shortly. But a couple of things to also point out is there's this junction here where you see pectinate muscle and you see sinus venerum, and we refer to that as the crista terminalis, and that's just that ridge where you see the dividing point between rough endocardium and smooth endocardium. You'll also see the opening for the coronary sinus. The coronary sinus is going to be a dumpage of all the venous blood from the coronaries back into the heart, and that happens back in the right atrium, basically just recycles the blood. And then the other structure is the Fossa Ovalis. So that's a remnant of that foramen ovale I was talking about, where you get a communication between the left and right side. So another remnant structure, so if we're talking about fetal circulation now, is we have the foramen ovale, which is communication. The other communication was known as a Ductus Arteriosus, and I'll show you on here, is a communication between the pulmonary artery, the pulmonary trunk, and the aortic arch. And so usually what you'll see in adults is there's a ligament that we refer to as the ligamentum arteriosum, as you'll just see that holding the aorta and the pulmonary trunk together. So be on the lookout for that. And then in the right atrium, you'll see a depression, which is the Fossa Ovalis. And ideally that should be closed and not patent, but sometimes that's not the case. So what did those, what was the point of those structures is during fetal development, the lungs aren't working. And it's not because they can't work, it's just they're not getting oxygen. Basically all the oxygen is coming from the mother's blood. And so when we receive our oxygenated mother's blood, it's basically gonna follow in the normal path and dump into the atria where typically our deoxygenated blood would go. So we don't need it to go to the lungs. We're gonna try to bypass the lungs altogether. So the first path of getting this oxygenated blood from the mother is we'll send it over to the left atrium, because we want it to go into the left ventricle and pump into the body, right? Now, some of it's gonna go through there through the foramen ovale and basically bypass the right ventricle, ideally. And that's where that sinus venerum comes into play, because we want to keep that blood as funneled towards that foramen ovale as possible. Now, it's not perfect, so sometimes blood's going to actually get into the right ventricle. More importantly, we're going to get systemic blood that's deoxygenated, basically going from the right atrium into the right ventricle. So the other way to get past that is we need to bypass the pulmonary circulation, because our left and right pulmonary arteries are going to be very narrow and basically stenotic. So you shouldn't be getting any blood flow to the lungs until the fetus has been born. So it's going to bypass that through this Ductus Arteriosus. And so that's how we can keep getting our blood. And it's basically a mixed composition of blood
So you don't have pure oxygenation in the fetal circulation. It's going to be a hybrid because we need to bypass the lungs completely. So once we lose oxygenated blood in the system and it gets in the atrium, it's going to mix with the oxygenated blood from the maternal surface. The maternal's artery. So what we'll see is after birth, pulmonary circulation is going to open up, and this pressure is basically going to allow the left side to become its normal pressurized system, and that's going to force the foramina vale closed, and that becomes the fossa ovalis. It should be. Sometimes you get those as a patent structure, and you get what's known as a heart murmur. And then eventually this ductus arteriosus is just going to narrow and become fibrotic, and it creates that ligament. So those are just remnant structures. Just like anything in the body, when stuff's supposed to go away, sometimes it doesn't. So I already mentioned you can get the patent foramina vale in adults. Usually it's a non-issue. You can live with it. It's just like a heart murmur. If it's really big, then sometimes you have to go and correct it with surgery. You can also get a patent ductus arteriosus sometimes, and that's something they can fix with surgery as well. So just be mindful of those two structures. Ductus arteriosus becoming ligamentum arteriosum, and the foramina vale becoming the fossa ovalis. So that is an objective you might be tested on. All right. All right, so that's everything with the right atrium. The right atrium had a lot going on. It was pretty exciting. Now we get to the right ventricle, which is a little more standard. So this is pretty much just a muscular component, and a lot of that musculature is going to be this rugged, what we refer to as trabeculated look. And so it's aptly named Trabeculae carne. So I always just think carne, kind of like steaks. So it looks like little slices of steak in the web or something. I always associate my anatomy terms to food. So trabeculae carne is all that rough muscle that you see in the ventricles, and that's the same for the left and the right. Now in the atrium, remember that was pectinate muscle, so try not to confuse the two of those. As you go towards the pulmonary arteries, so remember blood's going to come in from the right atrium into the right ventricle, and it's going to go towards the pulmonary trunk. And so what we have there is this Conus Arteriosus. Again, that smooth muscle helps funnel that blood, and so you'll see as you get closer to your pulmonary trunk that the endocardial layer smooths out, and we refer to it as Conus Arteriosus, and it helps kind of funnel the blood so that when you pump, it goes straight towards the pulmonary arteries, puts less strain on the valves there. The dividing point between the rough patch and the smooth patch we refer to as a supraventricular ridge. It's really kind of a pain. Nothing I would ever really seriously tag, but it's something to be mindful of. Just like in the crista terminalis in the atrium, it was kind of a dividing point between rough and smooth. We have one in the right ventricle. A couple other things to make note. So the valve that we have on the right side is called the tricuspid valve. So these are a type of AV valves or atrial ventricular valves because they go from the atrium to the ventricle. So these base of the valves, these are kind of like if you think about somebody going windsailing or flying a kite. So the goal is to use the pressure to kind of rein everything in. And that's exactly what happens. So what we have in these AV valves is we have these leaflets that have these things called chordae tendineae attached, which literally is heartstrings. So if you talk about heartstrings, chordae tendineae, and they're attached to these structures called papillary muscles. And so when the heart contracts during systole, the blood pumps out, and those valves are little thin leaflet valves. They want to push out back into the atrium, but because of those chordae tendineae holding tightly to these muscles, which are actually going to contract during the contraction phase, they pull back and basically create this like kited sail effect. It's kind of really neat to see. But we call this one in the between the right side tricuspid because it has three cusps or three leaflets. So you have a septal, anterior, and posterior. Now in the right side, we'll see mitral valve or bicuspid valve, which only has two, anterior and posterior. Now the other thing to be mindful of is the only thing, this is only found in the right side. It's called a septal marginal trabeculae, or more aptly, not aptly, but commonly referred to as a moderator band. I'll take either both are common terms. And the moderator band is actually a band you'll see coming from the interventricular septum, so we mentioned that, and going to the papillary muscle. Typically it's the anterior one. And so what you'll see is just this like papillary-like structure. It looks kind of like a little finger coming from the wall to a papillary muscle, and that's your moderator band. And it's really good to see on a, you can get an image of a heart. It's just, they come in varying sizes, and usually in labs we have students that'll accidentally cut it because it's tiny and it looks like a chordae tendineae, and then you have really big ones. So it's really exciting when you can see one. So just be on the lookout. But I always tell people papillary muscles should be attached to chordae tendineae. If you see something that looks like a papillary muscle, but it just seems to be coming from the wall and going to another muscle, that's your moderator band, or your septomarginal trabeculae. And it's only found in the right side, and I'll go into that when we get into the contraction of the heart. All right, so now that's all the right side. We've pumped our blood into our lungs. You can refer back to Viscera 1 or Lecture 2 on the thorax for how that works. Now blood's coming back. Let's talk about how it goes into the left atrium. So we have 4 pulmonary veins. So if you remember from our lungs, the pulmonary veins were kind of just this chaotic mess, and eventually they coalesce into those 2 on the left side, 2 on the right side, and then they'll merge into the left atrium there. So the entire left atrium is smooth. However, there's also an auricle on the left side that does contain pectinate muscles. So if I said, is there pectinate muscles in the left atrium? Technically there are, but they're just not as prominent. So if you were to open those up, it would pretty much predominantly be smooth. But all the pectinate muscles on the left side are contained in the auricle. They're in the auricle and the actual atrium on the right side. Now, if we're going to pass blood, there's really nothing else in the, you don't typically see the Fossa Ovalis on the left atrium. There's really not a whole lot of unique structures over there. It's just kind of like a dumping ground for a lot of oxygenated blood. So after that, we're going to go through the mitral valve, which is another atrioventricular valve, AV valve. And it's also known as bicuspid, so you can hear both terms because there's 2 cusps. Works identically as the tricuspid, where we have our chordae tendineae, our leaflets, and our papillary muscles, but we only have an anterior and posterior cusp. But they're pretty big cusps. So if we're thinking pathology-wise, the mitral valve tends to be more prone because of a lot of the pressure that it's under, more prone to pathology, things like calcification builds up and regurgitation. So a lot of times you'll see mitral valve prolapse, which just means it's not working and blood's being able to go back into the left atrium because maybe the chordae tendineae aren't functional, or the heart has gotten so big that the closing mechanism isn't functional. So just be mindful of that. And the same with the aortic valve when we take a look at that. So let's take a look at a heartbeat. So we already talked about diastole and systole, right? So in diastole, the left ventricle is going to relax, papillary muscles are going to relax, everything's just kind of chill. There is really, the only pressure is coming from passive pressure in the atrium. And this is going to let the leaflets of the mitral valve, which is just this, they're really flimsy things. They're just this connective tissue that just, I mean, you can kind of just push them out of the way. They don't look like anything spectacular if you ever see these. When they're fixed, they actually look a little more interesting than when they're fresh. But the blood in the atrium just kind of falls into the heart. You do have pressure, so you have pressure from the lung circulation stuff putting in there, so it's not just a complete passive process, but it just kind of dumps itself into the ventricle, okay? And then the heart gets the message to contract. So the left ventricular walls, interventricular septum contracts, the papillary muscles contract, everything is contracting and just holding tight. And we've already talked about that concentric nature, so the heart's just going to kind of do that spin, ringing out thing. And what's going to happen is papillary muscles are going to like button down the hatches, as they say on the ship, and brace for impact and hold those chordae tendineae intact. And then as the blood tries to push out, it's going to push against your mitral valve, but they're being held in tight because of the chordae tendineae. So it's like, well, the only way it can go is through the aortic valve. And we'll see why, because the aortic valve is actually very, those leaflets and cusps are actually also very loose and easy to kind of disperse. So we have pulmonic and aortic valves are referred to as semilunar valves. And the reason for that is if you were to look at a cross section of that, they kind of look like cups, so like half a moon. But they're both the same type of valve. They both have 3 cusps to them, the pulmonic and the aortic. And during diastole, they basically allow blood to just fill up. So they're basically these cups along the edges of the vessel, and as blood just would want to pool back in and the heart is relaxed and not under external pressure from the ventricle, they just fill up like little cups, and you can kind of see them there. And that filling up process is what closes the valve. All right. Now, sorry, I'm losing my voice on this one. So the difference between the pulmonic and the aortic is, first off, they both have a left and right cusp. And so the only difference is one has an anterior cusp and one has a posterior cusp. And the way I always like to remember this and which one has which is the aortic is going to have a posterior cusp and the pulmonary is going to have an anterior cusp. I personally just like that it's the saying, every heart needs a PA, and I think a lot of people can relate to that. So if you're talking about the aortic, you got a left and a right. A is aortic, so you need a P, posterior cusp. And then if you all the pulmonary, you have a left and right. It needs a well, pulmonary is a P, you need an A, so there's an anterior cusp. And so you can kind of see here, if this is our anterior, this is our posterior cusp on those ones. The interesting thing on the aortic valve is the left and right cusp have the corresponding coronary arteries. So when they fill up during diastole, the blood can actually just go and disperse through the coronaries as well. Now, those things are not sturdy, and so during systole, what happens is the heart contracts and pressure just pushes out from underneath. And just think about if you had a bag filled with water and you came up from underneath and smacked it, all that water is just gonna leave. And the bag's just kind of like flatten out and do nothing spectacular afterwards. And that's exactly how these semilunar valves work. So they just get pushed open, no resistance, nothing. The AV valves are holding tight with their chordae tendineae, and then the semilunars are basically just going to allow the blood to just pass right through. So then diastole occurs, blood starts to want to come back towards the lower pressure system, but those semilunar valves are going to fill up like cusp, close, and blood's not going to go back into the heart, ideally. Now again, we can get things like regurgitation or calcifications where these won't close properly, but hopefully that's not the case. So I'm sure we've all been in a position where we have to go get our physical done, and they always do the heart sounds on there and try to listen to your heartbeat. So typically you can do the generic one, which is Erb's point, where they kind of do it at third intercostal space, and they try to do should listen to all the heart's sounds, but you could singularly isolate each projection for valve. So if you wanted to test the person's valve and see like what valve might have dysfunction, so I already told you mitral and aortic can have dysfunction sometimes, then depending on where the projection of that sound will be, so aortic valve is going to be on the second right intercostal space, right? So that sound of the blood coming out and filling up is going to project here. So if you wanted to listen for that, you just kind of go just to the right of the sternum in the second intercostal space. Pulmonary valve is on the opposite side of that. Tricuspid is going to be the fourth intercostal space, and mitral valve is on the fifth, so right about at the end, the apex of the heart, where we kind of saw the projection of that. That's going to be the projection of your mitral valve. So you can kind of singularly isolate those and listen to the auscultations for those. So that's just something to be mindful of. Some of you will do clinical practice where you test that out, and it's just kind of cool to like practice the various sounds. But ideally what you're listening for is the valves closing and opening, and those sounds will kind of project in various areas. So here's kind of a nice little visualization of how that sound projects, so you can kind of see where you're listening for and why you're listening for those particular regions. So again, you can singularly isolate each valve. Usually when you go to a physical, they just kind of do the Erb's point spot where they can be like, okay, all the valves sound good, but if they want to go a little bit deeper, that's an option for them. All right, let's take a look at the vasculature of the heart. So even though the heart is essentially a glorified blood vessel or a large blood vessel and a pump, it still needs to receive its own blood on the outside, otherwise it's under risk of becoming ischemic and suffering from a heart attack. So the primary areas of getting vasculature to the heart is the coronary arteries. So looking at that, and those coronaries are going to return blood via the cardiac arteries. And so those are all going to be spread out. So if you look at the surface of the heart, you're going to see these arteries kind of spread throughout, arteries and veins kind of spread throughout, but there is method to their madness. So let's take a look at the coronaries first. So I mentioned that they branch off the aorta, and the easiest thing is they branch off the corresponding cusps for the aorta. So the right cusp has the right coronary ostia, which is an opening or exit point. So the blood for the right coronary is going to go on the right cusp. The left cusp has the left ostia or left opening for the left coronary. So the right coronary is going to rise from that sinus, the aortic sinus in the right side. It's going to give off, so you can come here and you'll see a right marginal vessel, which is going to predominantly supply your right-sided ventricle and atrium. And then it's going to wrap around, and in most hearts, it creates what's known as the posterior interventricular branch. And this is going to descend along the posterior interventricular septum that you can kind of palpate. And that's the septum is the point that divides the left from the right ventricles. There's also going to be a left ventricular branch, which comes off of the right side. It's kind of like a remnant. You can sort of see it down here. That's nothing I really would quiz you on. The biggest ones are knowing the marginal branch and the posterior interventricular branch. And that's going to supply predominantly all your, a big portion of your right versus your left ventricles on that side. So the other thing to mention is, well, I'll get into heart dominance shortly. But let's take a look at the left coronary and its divisions. So left is interesting because it starts in the left cusp and it almost immediately splits. So it's going to split into a circumflex branch, which is going to wrap around to the back side of the heart. And it's going to split into an anterior interventricular branch. Now, a lot of people will call this left anterior descending, which is a bit of a misnomer, but it's one of those things I don't penalize people on because it's so common in the vernacular that it's just not even worth struggling with people. But so if you're used to calling this an LAD, whoops, right there, which stands for left anterior descending, that's fine. It's technically the anterior interventricular branch. And the reason why is if it's a left anterior descending, where's the right anterior descending, right? But I know you guys have seen some weird quirks like that in anatomy where they'll call something a longus or a brevis without a longus, and etc. So it can be funny sometimes, but unfortunately, that's less common than often than not. So that one's going to supply your anterior side left and right ventricles. The circumflex is going to come around and then give a left marginal, which is going to be left ventricle and left atrium. And then it's going to come back and meet up with the right side of the heart in the posterior interventricular groove. Okay, so your main branches are left coronary, anterior interventricular branch, and you can see where that's just, so where that branch is, that's the septum, and it's identical on the posterior side. The right coronary is going to lie in that septum. You'll get your circumflex coming around, your left marginal, and then the circumflex will just basically continue on. So here's a nice little labeled diagram. This is just a little thing to go over, so I'll just click through these. But you have your right coronary here. Your marginal is going to be on the side. Posterior is kind of showing you that's in the back. Is that long one going right down the back of the interventricular septum. Left coronary up there, coming out of the left cusp. The first branch we'll get is anterior interventricular / LAD, and that's going through that interventricular septum. You'll get a circumflex right there, which is going to wrap around, and then you get the marginal, left marginal. So those are the ones I really want you to focus on as far as studies go. Now, I did say I want to get to variations. So if you've ever heard the term of something being a right-dominated heart versus left-dominated heart, the variations that you see, most hearts tend to be right-dominated, but that information is kind of biased, and it really just depends on what population you're looking at. But when we talk about variations or dominating of the heart, what we're referring to is which artery is supplying that posterior interventricular groove. Typically it's the right artery, so you call hearts right dominant. Sometimes it's the left, so it's a left dominant heart if that circumflex goes down and supplies that posterior interventricular groove. And then sometimes it can be codominant. So both of them come around and your right and left equally provide that circulation to that posterior interventricular group. So just be mindful of that. It's one of those things in anatomy. Most hearts tend to be right dominated, but if you were to ever see those differences, just know that it's not uncommon either. All right, so let's take a look at the venous drainage of the heart. So everything's eventually going to drain into the coronary sinus. So I already kind of mentioned it. But the only coronary veins I really want you to focus on are these three here. There's a great cardiac vein, middle cardiac vein, and small cardiac vein. And the nice thing is they all associate with some of those coronary vessels we already looked at. And so that's probably the easiest way to know them. But ultimately they're going to drain into the coronary sinus, and the coronary sinus is going to drain into the right atrium and basically get that blood oxygenated again. So the great cardiac vein is going to be sort of the start of the whole cycle. We'll see that on the LAD side, or the left anterior descending / anterior interventricular artery. You'll see your great cardiac vein come up. And then as soon as it gets up around the interventricular groove, the atrioventricular groove, the groove between the atrium and ventricles, that's when it becomes starts to become the sinus. The middle cardiac vein is going to be the vein that comes back from the posterior edge. So we saw the posterior interventricular groove. It's going to come up and feed into that coronary sinus. And then the small cardiac vein, you'll actually see arising on the opposite side, right where the right marginal artery should be coming. And it's going to kind of loop in and hit the coronary sinus just before it enters into the heart. Now, typically, if I was going to tag coronary sinus, I like to do it on the back side. Right around here, you can see a nice big venous structure. It looks like a vein. It's just sort of attached to the heart. But just be mindful of that. Those are the three really to worry about. There's a lot of collateral branches of arteries and veins, but you can go down a rabbit hole with that. We'll just get to the basics, especially for clinical studies. Only these ones tend to really matter. Okay, now let's look at circulation. So the most important thing for the heart is to have controlled rhythmic contraction. And the way that's done is through these pacemakers. So we start up here with the SA node, which is the sinoatrial node. And it's going to be located just underneath the vena cava over there. You can see that dot. So what this does is it has specialized cardiac muscles, and it kind of gets the pace of the heart going. All right, so it's going to initiate the impulse and then send that to the rest of the heart to make sure everything contracts in a simplified, simultaneous manner. And so first thing it's going to do is transmit its signal to the AV node, the atrioventricular node, which is located just above your tricuspid valve here around the atrium. And then those fibers are going to split into the interventricular septum. So this is the good view of our septum. And we're going to create the interventricular bundle of His. Okay, and that's just going to be this big bundle that's eventually going to split into left and right Purkinje fibers, which are going to spread out and innervate the entire wall. So the goal is to get this completely in sync. So SA node starts the pulse, goes to the AV node. We get the bundle of His, which is going to split, create our Purkinje fibers, which are going to go out to the entire wall of our ventricles and to our papillary muscles. Remember, we need those to contract as well. And that's going to control the contraction in a nice uniform pattern. So as long as those are working fine, everything contracts in sync. And like I said before, I was going to talk about the moderator band, why that's important, is we have a lot more, I shouldn't say a lot more, but we have more leaflets in the right side. So remember, we have an anterior, septal, and posterior leaflet for the tricuspid valve. The moderator band just creates a shortcut so that those Purkinje fibers can get to those papillary muscles in the same time and not have to worry about being delayed. So that's sort of the reason why we see it on the right side, whereas the left, it's only the two cusps that are in control. We don't necessarily, they're easier to regulate, I should say. All right, as far as innervation of the heart, so a lot of your autonomic fibers are going to be found within the cardiac plexus, which is, again, sort of in your anterior mediastinum. Just up here, you'll see a lot of network. If you're doing the dissection for these, it just looks like a network of fibrous tissue, but it's all autonomic nervous system. So this cardiac plexus is going to be made up of parasympathetic and sympathetic fibers. Now the parasympathetic fibers will be coming from the vagus. Sympathetic fibers are going to be coming from the sympathetic chain, and then they create this plexus, which is going to be distributed through the rest of the heart. Okay, it's going to control the coronaries, it's going to control our valves, it's going to control the SA node. Everything comes from that coronary plexus. And apparently I didn't have it in here. So the main thing to think about with this is it's going to be slightly different. So if we get, and this one's a little more straightforward, if we get sympathetic innervation from our cardiac plexus, what that means is we're basically going to start increasing our heart rate, right? So we're going to stimulate our SA node, start increasing the heart rate of our AV node and Purkinje fibers. If we get parasympathetic innervation, we lower our heart rate. And so just be mindful of those, where those fibers are coming from and ultimately where they're leading. A couple of clinical correlates for this before we kind of call it a day. So cardiac arrhythmias, heart attacks, anytime you get poor perfusion. So in cases of long-standing atherosclerosis, what can happen is those coronaries can start becoming blocked. And when they do get blocked, the distal areas start to suffer. And eventually that tissue starts to die. So we talked about ischemia and infarcts, and that can happen in a heart just as well. In long-standing cases, so a heart or dead tissue is not necessarily guaranteed for a heart attack. Sometimes it will start to heal. And so here in this image, you can actually see anything from an acute couple hours old infarct to a fibrotic one. This is a very fibrotic one. So the thing to know is likely this wasn't the one that, I mean, eventually it could have been a lethal infarction, but the body had to have time, about 2 months, to heal this to become fibrotic tissue. But you'll notice that in this case, we lose the elasticity of the heart. So all this nice organized system from those conduction fibers that we looked at becomes disrupted when the area of the heart becomes dead and replaced by fibrous tissue. So typically, if we do happen to get an infarct in a particular area or cause a disruption in our conduction system, that's what's known as arrhythmia, and those can be lethal. And sometimes what happens is it's just a sudden arrhythmia. We don't see any sign that the infarct actually happened because it was so rapid that we don't see these like acute signs. It actually, you look at the heart on a autopsy and it just looks like, oh, I don't see anything, so maybe it was an acute infarct. But a lot of times in those cases, there's long-standing changes that indicate that the patient may have suffered from chronic heart disease. Other things to consider is cardiomyopathy, which literally translates to heart muscle disease. We divide these into three main types. There's dilated, hypertrophied, and restrictive. And they basically just indicate what the muscle is doing. So in dilated, the heart chamber is actually dilated, and eventually it's going to compensate by making the wall a little bit thicker. But usually what you see, particularly in alcoholics, is a big dilated chamber, which makes the heart floppy and hard to contract. Compare that to, and this is over here, a dilated heart, so you can see how big compared it is to a normal heart. Hypertrophied is anytime you have long-standing hypertension, the walls are going to get thicker and thicker to push more blood out because they're basically meeting resistance in the periphery. So your muscles get thicker, your ventricle gets thicker, which limits the amount of blood that's being filled, and the heart tries to compensate for that, and unfortunately it can only do it for so long before it expires. And the restrictive is usually associated with some specific diseases like amyloidosis. Something gets deposited into your heart muscles, and that pumping action basically gets inhibited. So those are just a couple of diseases that you can get. But other than that, that is heart. Thank you for joining me for the thorax unit. I think the thorax cavity is pretty fun, as is the abdominal, but let me know if you have any questions, and thank you for your time.